All right, so next on our agenda is a chance to learn more about personalized lung cancer care. For us to see what the current options and new treatments are on the horizon, we're very pleased to have with us Jürgen Wolf, who is Medical Director of the Center for Integrated Oncology at the University Hospital of Cologne. After his presentation, we hope to have some time for questions. Please submit them on Slido. All right, again, this is time permitting. With that said, I'll hand it over to Jürgen. Thank you so much.
Thank you for the kind introduction. Hello, everyone. It's a pleasure to be here and to speak about personalized lung cancer care. So these are my disclosures, and these are the key messages of my talk. Lung cancer is not one disease. It consists of multiple subtypes. And treatment is defined by these subtypes, means by tumor biology. Targeted therapies and immunotherapy have transformed outcomes substantially. New treatment strategies go beyond classical targeted therapy. And the main challenge today may not be science, it may be access of all patients to these innovations. So in advanced lung cancer, and this is shown here exemplarily for lung adenocarcinoma, we can distinguish today two fundamentally different treatment strategies in systemic therapy. And this is symbolized here in the gray segment, are patients without classical driver mutations, and in the green segment, patients with KRAS mutations. For these patients, we have no first-line targeted treatments available, and the standard treatment for these patients is immunotherapy with or without chemotherapy. And then we have these colored segments. These are patients with classical driver mutations, EGF receptor Al-Cross, and so on. And here in first-line, targeted therapies are standard, and these are mostly so-called TKIs. These are tyrosine kinase inhibitors, small molecules working after the lock and key principle, orally available, mostly better tolerated than chemotherapy. And this means that the right treatment starts with the right diagnosis. And what you can see here, a little bit simplified, is a diagnostic workflow, starting on the left with a biopsy, then the pathologist who is confirming the diagnosis, DNA, RNA extractions, and these new technologies like next generation sequencing, finally detection of the mutation and the molecular or organ tumor board. So means no testing, no personalized treatment. But what I also want to mention here is that there is an increasing complexity of technology, which needs more and more a high degree of specialization. So nevertheless, targeted therapies have transformed patient outcomes. Do not want to show you a lot of statistics, but just one patient example. This is one of my patients. So stage 4 NSCLC was already diagnosed in 2012. In 2014, the new ROS1 fusion was first described, and we detected this fusion in this patient. And we started treatment with a so-called first-generation inhibitor, the crizotinib at that time. And there is an impressive response. You can see from left to right the response in the PET images. And this response is ongoing still in 2026. So this means that more and more patients now live for years with a good quality of life, and we could never imagine that the course of disease in advanced NSCLC, let's say 10 to 15 years ago. But of course, let's come back to daily reality. The central challenge is resistance, because in all patients with targeted drugs, resistance is inevitable. So this is a summary cartoon, a little bit complicated, and I have heard that I do not need to go into the detail here because Roxanne in the next talk will go into the detail to explain these mechanisms. So I just want to introduce with this slide two wordings. So one is on-target mutation, means resistance is because the receptor itself is changing, and the other is off-target mechanism. This is when another oncogene becomes active in addition to the initial oncogene. So I have some technical problems. So these are the current strategies to overcome resistance. So targeting on-target resistance is realized by the development of next generation TKIs, targeting of off-target resistance. An elegant approach is TKI combination therapy. Then we have other new combination approaches and totally new therapeutic concepts. Just want to give you a few examples for these strategies. So this is really a success story. The characterization of on-target resistance as a prerequisite for the development of next-generation inhibitors. Just want to give you one example here, the ALK inhibitors. And you see in the table on the left, crizotinib, the first-generation inhibitor. We had the second-generation inhibitors, alectinib, brigatinib. We have the third-generation inhibitor, lorlatinib. And already in clinical evaluations, the neladalkib as fourth-generation inhibitors. And look at the overall response rate, ORR. There are no striking differences, 60 to 70 to 80%, not so different. But look, marked in red, at the median progression-free survival. This is the time where the tumor is under control by the TKI. And this is progress. Around 8 months with the first-generation inhibitor, up to 3 years already with a second-generation inhibitor. And we do not have the results so far for the next generation inhibitors, but they will be better. And it's really a must today to treat the patient with the best available inhibitor already in the first-line situation. Like our US colleagues say, best drug first. So a second example for off-target resistance. This is also one of my patients. At the first diagnosis of NSCLC, he was around 60 years old. He had an EGF receptor mutation. For 12 months, good response to osimertinib. After 12 months, you see the first CT scan here, massive relapse, symptomatic relapse. And we did a rebiopsy, and in addition to the initial EGF receptor mutation, we found a MET amplification. This is the activation of the second oncogene. Then we treated with a combination EGF receptor and MET TKI, and you see after already 3 months an excellent response. And also this response is still ongoing in 2026. But of course, this was an off-label treatment, and we had to ask the health insurance company for reimbursement. Third example, the new combinations. And so now more and more TKIs are also combined with chemotherapy. What is the idea? It's not a non-brainer. The idea is to reduce with chemotherapy the genetic heterogeneity, or to eliminate, to say it a little bit more easy, to eliminate resistance mechanisms in the beginning. This is a phase 3 trial comparing in the first-line treatment osimertinib alone with osimertinib and chemotherapy. For those of you who are familiar with these curves, you see on the left side, of course, the superiority of the combination. For those of you who are not so familiar, the message is quite simple. On the right side, osimertinib alone, progression-free survival around 17 months, and 8 months more with a combination therapy, but at the cost of definitely higher toxicity. And I think this is a case for shared decision-making. The doctors and the patients here have to balance together efficacy, toxicity, and patient preference. So if a patient is older, he's afraid of toxicity, maybe you consider TKI immunotherapy. But on the other hand, if a patient is young, good performance, that's not so afraid, you can start with a combination therapy. So I have talked now nearly only about TKIs and next generation TKIs, also about combination with chemotherapy. But there is now a really substantially increased treatment landscape with a lot of new drugs, which are, and I think this is also important, which are not exclusively developed for mutation-positive lung cancer, but also for non-oncogene-addicted lung cancers. These are antibody-drug conjugates, bispecific antibodies, totally novel mechanisms you have never heard about, and of course also new immunotherapeutic approaches. And again, just a few examples. Antibody-drug conjugates, heavily discussed within the past 2 to 3 years. Very simple, the cartoon on the left, it's an antibody, and it's linked to a chemotherapeutic molecule. In reality, several chemotherapeutic molecules, symbolized here by the red point. And there are different target structures. So the idea is very simple. The antibody brings the chemotherapy to the tumor. And he recognizes a target on the tumor. And on the right side, the table just showing you that an ADCs against a lot of targets are developed. For instance, a TROP2 is a target which is expressed on mutation positive and mutation negative lung cancer. So this dichotomy between lung cancer with mutations and without mutations somehow disappears with the newer developments. So ivonescimab, an example for a new bispecific antibody. He has two specificities, one against PD-1, like the PD-1 immune checkpoint inhibitors, and one against VEGF. This is a molecule triggering formation of blood vessels in tumor. We call it angiogenesis. And surprisingly, this bispecific is also active in EGF receptor mutated patients, also in mutation positive. And as you see, again, a phase 3 trial, again, you see a difference in the curves. These are patients with progression on EGF receptor TKI therapy, and the investigators here compared chemotherapy only against chemotherapy with this ivonescimab. And you see it's superior. The absolute benefit is modest with 2 to 3 months only, but each advantage is clinically relevant for the patients. And now a question which is often asked by the patients: what about immunotherapy in my mutation-positive lung cancer? You all know that immune checkpoint inhibitors do not work very well in mutation-positive lung cancer. And one of the most frequent questions to me is, what about vaccination? So what is vaccination? Vaccination is a more than 100 years old idea or dream of the immunologists, and it's just to activate the patient's own T cells against his or her tumor. And there are different technologies. You can inject cells, you can inject proteins, you can inject other constructs. And one of the most modern approaches, of course, is mRNA vaccination, but the principle is the same, the idea is the same, and this technology was developed during the COVID pandemic. And but I want to, and there are a lot of trials, and we are also participating in these trials, but I have to say, so far there is no proven efficacy of this approach. There is not a single approved vaccination approach. So I personally am very skeptical. Why? Because the tumor has thousands of strategies to escape the attack of the immune cells. So this is an approach I personally think is more promising, but also more toxic. These are CAR T cells. What are CAR T cells? So on the left, you see a T cell. In the middle, you see an antigen receptor. This antigen receptor is brought into the T cell. It's a complex procedure. So this T cell is genetically modified, and the receptor in the T cell now brings this T cell to the tumor. This has been very successfully developed in the lymphoma field, a lot of approved drugs, but it's in the very beginning with solid tumors. There are a lot of technical problems. I do not have the time to go into the detail, but there's a very dynamic development also of next generation technically improved constructs, and it's a rapidly evolving field with an increasing clinical activity, in particular, for instance, in China in EGF receptor mutation patients. So I summarize what I have shown you to the therapeutic landscape. It started with the detection of the driver mutation. We had the first generation inhibitors and then the next generation inhibitors, and now a bunch of new therapies. So why are not all patients benefiting from these therapies? And I think this is not a scientific problem. This is a system problem. There is a limited access to molecular testing across Europe. There's an increasing complexity of diagnostics, which needs a high degree of specialization, as already mentioned earlier. And there are fragmented healthcare structures. And this is not on the slide, but has discussed this morning. There is a very different willingness to pay for such expensive therapies in European countries. So this is the situation somehow. We are overwhelmed by a huge wave of innovation with a lot of new biomarkers, a lot of new drugs. And the question is, or the challenge is, how can we as a community keep up? And I want to highlight 2 strategies which, in my opinion, will become more important: networks with shared workflows, professional networks, and patient participation. What is the idea of shared networks? It's a shared workflow between specialized centers, and the responsibility of these centers then is molecular diagnostics recommendation, molecular tumor boards, complicated trials, and so on. And the responsibility of the regional network partners then can be therapy, because therapy mostly is not so complicated, and this could be a chance for patients to be treated close to home. And I want to show you how we in Germany have realized this idea. This is the National Network Genomic Medicine Lung Cancer, founded in 2018, supported by all health insurance companies and the German Cancer Aid. And we cover now 2/3 of patients with advanced lung cancer. And there are around 30 specialized centers and around more than 600 now regional network partners, community hospitals, and practices. And this is exactly the workflow I have shown you on the previous slide. And from the beginning, we established a very close and fruitful partnership with patients. And here I have to emphasize Ziegenau. Ziegenau, translation may be precisely targeting, is a patient-led umbrella organization for oncogene-driven NSCLC in Germany with a lot of mutation-specific subgroups. It's now a nationwide network of patients with more than 1,000 members. And on the right side, you see the board of Ziegenau, 7 very impressive women, all suffering from oncogene-addicted NSCLC. 2 of them, Nicoline Erhardt and Heike Gantke, are here with us today. And on the lower part of the slide, you see an original slide of one of the talks of Ziegenau, which is a very active organization. And here the goals are summarized: to connect patients in mutation-specific subgroups, to improve patient knowledge on diagnostics and therapy, support access to experts and clinical trials, and more also to represent the patient voice in research and increasingly also in politics. And these networks are, and I'm really impressed by this activity, are also internationally very well connected. So I think this is the next level of networking. Beyond the professional network, the next network level are patients connecting across borders. And you are all familiar, I think, with these groups, the EGF receptors, the ALK positives, the ROS1ers, and so on. And I think these global patient communities, so consultation hours are often challenging because often the patients have more knowledge than the doctors. But I think these communities contribute unique knowledge from early trial insights to real world experience. And I think this shared knowledge leads to truly shared decision between doctors and patients on a new level. So with this, I come to my end. These are my take-home messages. Personalized therapy has fundamentally changed lung cancer care. Resistance remains a central biological challenge. New treatment strategies go far beyond classical targeted therapy. The main limitation today may not be science restrictions, but access to personalized cancer care. And I think that networks are essential to translate innovation into patient benefit. Thank you for your attention.
Thank you very much for your great presentation and for already flagging so many of the topics that we'll dive into deeper later today, including resistance to therapy, the importance of shared decision making and how we make those decisions, and the ever-present question of access. So I'm going to just switch over to Slido because we've got the privilege of having questions for you. Not to put you on the hot seat, but you are the expert. So we have Seamus Cotter here, who's asking, for those diagnosed with advanced stage lung cancer without a notable biomarker, is there no personalized treatment available? And a reminder to everyone to please submit your questions on Slido and upvote the ones you want to see answered.
So the question is, what are the personalized options for patients without classical driver mutations? Mm-hmm. So unfortunately, in the moment, we only have one predictive biomarker. So personalized therapy depends on biomarkers, and this is PD-L1, and it's also a prognostic marker. It's weak, so we still have to wait. And what is the problem? So these oncogene pathways, means mutation activation, signaling pathway, malignant growth, are relatively easy to understand and also for drug development. But the immune system is much more complicated, and so far we have not understand the tumor microenvironment. There are so many cell types, so many liquid factors, so many chemokines. We do not, and as long as we do not understand the tumor microenvironment, we really will have problems to develop efficient personalized therapies for immunotherapy.
Well, lucky for y'all, one of our next presentations is about the tumor microenvironment, so this is going to be really relevant to it. We have a question that's been upvoted a lot. So the small cell lung cancer field seems to be evolving rapidly. What new treatment approaches should we be aware of? You hinted at some in your summary slide, but tell us more.
So within decades, there was only one progress. This was the introduction of immune checkpoint inhibitors a few years ago. Median overall survival benefit, however, was modest with 2 months. And now we really have an exciting new development with the tarlatamab. Tarlatamab is a bispecific antibody construct which is directed against the T cell and the DLL3 on small cell lung cancer. So response rate in relapsed patients is around 30% only. But what we really see for the first time in small cell lung cancer are long-term survivors. So I personally, we have participated in the trials, and I have a young patient in the 4th relapse of his disease. He was treated with tarlatamab, and this was 4 years ago. He's still in a very healthy condition. This is really exciting. And also in clinical development are a large number of so-called ADCs. I have already introduced ADCs. Also promising response rate, but still early in clinical development.
Yeah, tarlatamab is the one I mentioned yesterday, which was, it received EMA marketing authorization, I think March 26th, for extensive staged relapsed small cell lung cancer. And I think from what I read, it has slightly different approvals in the US than it does here. So it'll be interesting to see how its use evolves with time as well.
So it's approved in the US already. It's a standard second-line treatment, but the trials recruiting in the first-line situation are recruiting extremely fast. So I expect the approval for first-line in the US next year.
That's pretty soon.
It's one of the first 10 to go centralized HTA assessment.
Well, there you go.
And the PICO was troublesome because what does extended
Relapsed, yeah.
Relapse means the indication was a big issue.
Yeah.
All right. Thank you for your contribution, Elena. The next question, so tricky. Are surgeries not available to stage 4 patients? Most of us are diagnosed at stage 4, young and relatively healthy. Must it only be just targeted therapy? We also have, I'm gonna voluntold the thoracic surgeon I know, and I've spotted over there, Cecilia, so maybe I can call on you as well to share an answer.
Maybe we should differentiate between multiple metastasis and the so-called oligometastatic disease. And there are strategies established and also recommended by the guidelines that if you only have a restricted number of metastases, that surgery may be possible. Today, mostly after neoadjuvant treatment, and I think this is one development. But besides surgery, and I hope it has become a little bit clear in my talk, it's not only the TKIs now, it's a combination of TKIs with a lot of other new drugs, even immunotherapy beyond the classical immune checkpoint inhibitors. And think that in stage 4 NSCLC, immune checkpoint inhibitors in patients without driver mutations have probably a cure rate of about 15%. So I never use the word palliative intention for treatment in these patients. And I think the development will be that we also can achieve such long-term controls in more and more patients with driver mutations already in the near future.
Thank you very much. I don't know if Cecilia, you want to add something on the surgery?
I mean, yeah, I agree with what has been said. It's important also to know that there are all these new real-world data and collection that done in, for example, in US, and there are also some centers in Europe where this approach in selected cases and with multidisciplinary team definitely will go ahead. It's important to read those guidelines now, the STS guidelines written on the oligometastatic disease approach, because it's not only surgery, but it's also radiotherapy has consolidative approach. So definitely more to come. If we are collecting, in this case, most probably real-world data will be crucial.
Thank you very much. One more question for you. So there's a question here that's saying, can you speak to prevention therapies in stage 1 and stage 2 cancers that have a genetic mutation? Healthcare providers only currently pay for stage 3 to 4 TKIs. I don't know if the question is clear.
Yeah, the question is clear, and it's a very difficult question because this is a gray zone question. There was an international meeting with around 40 participants, medical oncologists from all over the world, directly before ESMO, and this question was also asked, and there was not a homogeneous opinion because, for instance, we have these data now for EGF receptor mutation and for ALK fusion. There are trials ongoing with RET. There are trials starting now with HER2, but still there are no trial results. So normally there's no chance for reimbursement because these are very expensive treatments. But on the other hand, there is really a lot of evidence that this may work as it works in EGF receptor and ALK. So the question is very difficult to answer. It's a very individual decision. And it also depends on the reimbursement.
Yeah, that's all the time we have for these questions. Please join me in warmly thanking Jürgen for his great presentation.